a potential tool for crop improvement in chili pepper. Okay, you will do it, right? So, agenda of the presentation includes introduction, androgenesis mediated DS development, factors affecting DS development, and challenges and future direction. And I am also going to conclude what are the future requirements for the DH. Coming to introduction part, as we know, chili pepper is a globally important crop, valued for its culinary, nutritional, and economic significance. However, breeding program in pepper faces several challenges. So long breeding cycles, high heterozygous, and conventional breeding takes 6 to 8 generations to achieve homozygosity. And it also has complex traits like yield, pungency, disease resistance, and market demand for quality as well as uniformity. Okay, so now let's see what are these double haploid technology. Haploids are sporophytes having n number of chromosomes. Sporophytes having gametophytic chromosome numbers, that is n number of chromosomes. When it doubles the chromosome either by chemical method or by spontaneous doubling in the culture, you get a doubled haploid. The same set of chromosomes gets doubled so that both sides of the alleles having similar alleles, so that is why it is 100% homozygous. So that will have an immense application in the breeding programs. So the double haploid technology is a tool for reducing the generation for the breeding programs. You can see these are the major applications in breeding when you use double haploid technology. Homozygous lines are obtained within 1 year, so these can be multiplied and evaluated for quantitative traits much earlier in breeding programs compared to bulk or pedigree selections. So DH lines enable stable trait fixation and uniformity, supporting hybrid breeding programs for consistent quality and performance. Since they carry one allele of each gene, so mutation and recessive characters are expressed in the plant. So that is why you can select, so what are the recessive traits, dominant traits, all the characters are expressed in the DH lines. Because of the immediate homozygosity, because you fix the line within a single generation, so you will see the genetic variation among these lines are maximal because of the immediate fixation. So the genetic variation in early stages increases the selection gain. So these are the major applications of DH technology. So let's see how DHs are produced. There are 2 ways of producing DHs. One is in vitro method, the other one is in vivo method. The in vivo method includes having inducer lines, like for example in corn we have inducer lines, we can do it in the field level itself. So and then we also can produce DHs in vivo method by using parthenogenesis. But when it comes to in vitro methods, there are 2 major ways to produce DHs. One is by androgenesis, the other one is gynogenesis. Androgenesis is the production of haploid plants from the male gametophyte, that is by anther or a microspore culture. Gynogenesis is the production of haploid plants from the female gamete, that is unfertilized egg or ovary. So again, within androgenesis, there are different modes of androgenesis. You can see in the picture, we grow the plants and then we collect the flower buds. From that, we do anther culture. So right side, you can see the anther culture pictures where, so it depends on the crop, the mode of androgenesis is different. So when you culture the anther in the medium, you will see 2 ways, that is by direct microspore embryogenesis or indirect microspore embryogenesis. Whereas in case of direct microspore embryogenesis, you will get embryos directly from the anther culture. Example, you can see pepper. So once you inoculate the anthers, you get the embryos directly from anther culture. This method is called direct microspore embryogenesis. The other method is in some crops like cereals, you don't get embryos directly from anther culture. So you have to go through a phase called callus. You have to generate a callus first and then from that you have to regenerate. So that method is called indirect microspore embryogenesis. Example is rice. So you have to get a callus first. Embryogenic calli, that is very important. You may get callus from other parts of the anther as well. So you have to have a protocol in such a way that you have to get callus from the microspores. So then you have to regenerate the plant from that. That will become a DH1. So this is indirect way of producing DH. The other method is microspore culture. So where we isolate the microspores and then we culture them. We get embryos directly from microspores. So example in brassica, so you see the picture, it says this is canola microspore culture plate, where you can get embryos directly from the microspore. So this is 100% homozygous. So this three methods again depends on the crop or species. So the same protocol will not work for all the crops. So this is crop specific, and then again there are genotype specific protocols as well we need to develop. So these are the different stages of androgenesis in chili pepper. You see, we need to grow the plants under controlled conditions. Like you see, light, temperature. I'm going to talk about this a little later in detail. So and then once you grow the plants, we have to do, take the anthers and culture them in the medium. After that, you see the embryos, coming out from the anther culture after 6 to 8 weeks of time in the incubation period. From that, you have to go for separate the embryos and then culture them on the shoot elongation medium. The plant which you are seeing below, the DH plant in rooting. Why I am saying DH? Because it has to be haploid, right? So by seeing the plant, we can differentiate at that level itself whether it is haploid or doubled haploid. So this advantage we have in chili pepper, especially so that up to 40 to 50% of the embryos produced in vitro, they are spontaneously doubled. So that is the advantage we have in chili pepper, so that we can get doubled haploids within the culture without having colchicine treatment for doubling. So that plant, by seeing the morphology itself, we can differentiate between haploids and doubled haploids. So once you get the plant, so if it is haploid, you have to go for chromosome doubling, otherwise you can directly go for hardening. You can see these plants in acclimatization stages. So we have a different methods to acclimatize. The percentage of survival again is difficult work, but we have up to 90% survival rate once you harden the plants. And then you can see DS plants in fruiting stage. So you can see this is from the same background. You can see the different height, weight, color, everything. So the variations you see very good in the DS plants which you get from the single parent combination. So coming to the key factors, this is very, very important. So there are different factors which affects your embryogenesis. Number one is genotype. So there are some recalcitrant genotypes which won't respond at all. There are zero response. Some genotypes we may get up to 40 to 50% of embryogenesis. So that is why genotype plays a major role in producing DHS in chili pepper. And then donor plant growing conditions. So this is in our hands, but this is very important. So we need to grow the plants under controlled conditions with very good proper lighting, temperature. The plant should be the donor plant should not have any diseases while you collect in the flower buds. And then pre-treatment conditions. So I have a publication on this. You can go and Google it. So it is on pre and post culture treatments to enhance the embryogenesis in chili pepper. So which says after collecting the flower buds, you have to go for pre-treatment for certain period of time. Temperature is also controlled. And then once you culture them, there is post-culture heat treatment. Cold treatment followed by heat treatment. So then only, if it is a properly maintained, then only we will see embryogenesis results. Otherwise, it is very difficult to get embryos germinated. Next is bud and anther development. In microspore development, because it is germ cells, from the pollen mother cell, you will get meiosis starts, meiosis 1, meiosis 2. At the end of meiosis 2, you will get a 4-cell stage called tetrad. From the tetrad, so all the 4 cells will be released, and each cell will have n number of chromosomes. That is called uninucleate microspore stage. After meiosis, it will start mitosis. Once it starts mitosis, the starch accumulation starts, so that you won't get any result from that. So because we are deviating gametophytic pathway into sporophytic pathway into gametophytic pathway, you need to apply some certain stress and then take anthers or microspore at particular stage. So that is very important. So in my observations, late uninucleate and early binucleate stage of microspore responds better for our embryogenesis. Coming to the other important factor, which is culture medium composition. So this is also one of the important factors where the basal media combinations, hormonal combinations, everything is very important for the results to get. And then culture environment, where you keep those cultures. After culturing, you need to keep them under dark period for certain period of time, and then you need to transfer them into light conditions where the temperature, light, humidity also plays a major role. And then embryo conversion and survival. So whatever the embryos you get, I talked about 0 to 40 to 50% of embryogenesis. All the embryos produced may not convert into a plantlet. So there, that is also genotype based, and then the culture conditions also plays a major role in converting all the embryos you produce from embryo stage to the plant. And then chromosome doubling. As I already talked about the spontaneous doubling in case of chili. So In case of chili, it is 40 to 50% of the embryos convert, because it's highly unstable and sterile. So it automatically doubles the chromosomes and become doubled haploids. So that it will be 100% uniform. You need not to go for chromosome doubling. If it go for chromosome doubling, again the ploidy varies. So whatever the plants you are expecting in the field, it may not be uniform. So you will get all kinds of ploidy and then variations. So that is why getting chromosome doubling step is also very important. The concentration, duration of treatment is very important to get doubled haploid. So coming to the future challenges and the direction. So because it is highly genotype dependent, so we need to have a protocol in such a way that all the genotypes should respond. So in my earlier cases, some genotypes which they were not responding at all. Now we have developed a protocol in such a way that highly recalcitrant species also can produce embryos, but the numbers varies. If you inoculate 20, 30,000, you may get 1 or 2, or so maybe nil. So now we have a protocol. Now we get at least 100 DHS from the cross. So that genotype dependency is a major challenge for the researchers to get a DH. And then technical expertise is needed. So everybody cannot do, because you need to have a lot of experience in tissue culture, and then you need to have a facility also to get the results. And then cost and infrastructure. You need to have a sophisticated lab infrastructure for having growing the donor plants under controlled conditions, getting them into the lab. Lab, you need to have so many laminar rate flows. Other conditions are also required. So that is why cost and infrastructure you need to look into. And then when it comes to future direction, so wider adoption of breeding programs is much needed nowadays. And then combining with molecular tools and AI tools is the need for the hour, because we produce DHS, so breeder takes it and grows in the field, and then he will check morphological characters, so many characters he has to do in the field. Otherwise, if you combine the DS lines, whatever you produce through marker, so all the traits can be identified at basic D0 stage itself, so that will be very good advantage for the breeders to select which is directly required, and then he can go for F1 development. And then increasing efficiency of DS protocols in pepper, that is also required. So in India, majority of the companies work on the pepper. This is the basic method. It's like a tobacco for the transformation. So this is like a base chili pepper. So many companies are working on producing DHS in pepper. So that is why increasing efficiency is very much needed. So this coming to the conclusion part. So DS technology revolutionizes pepper breeding by reducing breeding cycle time and improving the genetic gain in a short period of time. Enable precise, uniform and superior hybrids development. Integration with modern genomics and AI ensures faster cultivar development. So this is very, very important. Can you see this photo? All these 5 plants we got from a single anther. Single anther. You can see the variation in the plant. So you can see pendant, upright, orange, red, everything can be get from a single anther. So that is see, so breeder will get all the 5 variations he is looking for within a single generation. So that is very much useful for the breeder. He can speed up, selection can be done very faster, so that he can develop a cultivar very faster. Thank you.
Thank
I
you
hope I'm on time.
for finishing in time. I just want to add 2 comments for everybody to know. I think you didn't probably mention. One is F1 pollen is used. That's why you get that variability.
Yes.
You have the F2 coming from those. So it's coming from not basically anther. Anther will be tapetal. So it should come from the various cells of the anther, and each one is different, because tapetal cells are diploid already, right?
Yeah.
So you have to get it from the microspores.
Microspores.
Second part is, I think, one is making inbreds faster, but DH is also a great technology to make recombinant inbred lines, so that in 1 year you have the population ready for doing genomic or any other trait inheritance studies, etc. Otherwise, it takes you 6 generations to get the inbreds.
Yes.
Right?
Yes,
so
yes.
that's another big advantage of DH. Thank you.
Yes.
One question.
Thank you for this, and I have a quick question. In terms of doubling of the chromosome without treating the colchicine, so what is the percentage of doubling it is actually happening?
It depends on your protocol. It varies. You can get 80% doubling also.
And as per your protocol?
I am getting 90%.
Okay, thank you.
80-90%. Because you see, one more advantage what I am doing is I am reducing the usage of colchicine nowadays. I am completely relying on spontaneous doubling, because colchicine is a very carcinogenic chemical. So I am reducing the usage of chemical. So by increasing the embryogenesis, so that you can get more plants, so that you can get 40 to 50% spontaneous doubling you are getting. If you get 100 plants, 200 plants, 100 will be all, no, it is doubled already,
Yes.
spontaneous doubled,
Yes.
so that you can save the chromosome doubling.
You need very good technicians. Oh yes, one from the industry. Percentage of sterility you get in the DH0 plants.
0? If it is a DH, why you get sterile?
That's what I didn't understand. Why do you get sterility? Why do you think there will be sterility?
If you talk about DH, it is 100% fertile. If you talk about haploids and double haploids, you will see if it is a haploid, it is sterile.
Means that you are saying it is that spontaneous chromosome doubling.
Yeah.
So, and you are saying it is a spontaneous chromosome doubling.
Yes.
And that even though that there is this I think that there is that chances of getting that sterility percent in the that DS0.
I think I suggest that you please discuss.
Yes.
With,
Okay,
See, if it is a CMS background, if it is an RF gene, then you can
yeah,
expect
if
something.
you are using yeah. Thank you very much. I think
So just to add a point for this, probably he was referring
yeah.
to
Please,
aneuploids.
please, everybody. Yesterday.
Probably he was referring to aneuploids. Do you see any aneuploids, or
Yes.
is it a perfect
Yes.
doubling?
Yes, when you go for chromosome doubling, there are chances of aneuploids.
Probably that will lead to sterility.
No, I am talking about only DH. If it is DH, there is no aneuploid there.
Yeah.
Okay, at the end you don't really find anything.
No.
Yeah.
Okay.
If it is a DH, you should talk about only uniform and fertile.
I think those plants are very vigorous and you can easily identify. Thank you very much. I think I request if there are questions, that bowl is empty. Please fill that question even for yesterday.
Thank
Thank
you.
you very much. Thank you, Doctor.
Thanks for the opportunity.